Hello, everyone. Welcome to Molecular Devices webinar on novel technologies for high-throughput ion channel assay development. My name is Xin Jiang. I'm the product manager for reagent systems at Molecular Devices, and I'll be your host today for today's webinar. Regarding the agenda, I would like to first spend a few minutes to introduce Molecular Devices, how we strive to stay innovative in serving the exciting field of ion channel research through continuous product development and also through exciting collaborations with partners like Assay. I'll be pleased to introduce the two guest speakers here today who will present first by Chiu-Fai Man from Cubist Pharmaceuticals on the development of an in vitro thallium flux assay for potassium channel agonist and identification. Followed by Chiu-Fai's talk would be Viviana Agus from Assay, and she will speaking on her recent project of combining Optogenetics and FLIPR system, where she used light to establish a high throughput screening assay for the CAV 1.3 channels. Toward the end of the session, like Brenda mentioned earlier, we'll have a question and answer session where you can submit your questions anytime during the webinar, and the questions will be answered in the order it's received. For the questions we cannot answer in the time frame, we'll keep the line open for another 15 minutes after the webinar, and you can keep submitting. And the questions will be answered by the speaker individually via email. The webinar will be recorded and posted on our website once it becomes available, and the link of the recording will also be sent to you in a separate email. So, Molecular Device has been a technology innovator serving the ion channel research market for more than 25 years. With a portfolio of marketing leading products covering all aspects of ion channel drug discovery process and also for the basic research. This slide shows you a number of the products we have for serving the ion channel market, starting with the gold standard conventional patch clamp systems, the Axon brand patch clamp systems, the amplifiers, the data and the software, and also a number of the automated high throughput patch clamp systems, including the Patch Express system, Ion Flux, and our flagship automated system in IonWorks Barracuda. Also, we have in the FLIPR system, which stands for Fluorometric Imaging Plate Radar, industry standard for high throughput drug screening. And the primary focus of today's presentation is around the FLIPRTETRA system, along with the reagent, which is optimized for analyzing ion channel activities on the FLIPR system. So particularly for today's talk, I'll be showing you the data collected with the calcium-6, which is a calcium fluorescence indicator, and the potassium kit, which is using the thallium as a surrogate assay for analyzing the potassium channel. So with that, I will start pass the ball to Chiu-Fai, who will start his first presentation. Chiu-Fai started his professional career at Warner Lambert in Ann Arbor, Michigan, after he graduated from the University of Toronto. At the Warner Lambert Research Facility, he worked in various therapeutic areas, including the atherosclerosis and inflammation. Earlier career highlights include a blockbuster drug, which ultimately led to the Pfizer acquisition of the Warner Lambert. He remained with Pfizer in Ann Arbor, prosecuting drug discovery activities like assay development and high throughput screening. In 2007, Chiu-Fai moved to Pfizer Cambridge, and in 2010 to Cubist Pharmaceutical. At Cubist, his primary research interests include developing and prosecuting assay for antibacterial, pain management, and host defense mechanisms. With that, I'll pass the ball to Chiu-Fai. Chiu-Fai, you're now the presenter.
thank you very much, Xin, for that introduction. And thank you all for attending this webinar. I'm going to talk to you today about my work in the development of the in vitro plate-based thallium flux assay to enable identification and optimization of novel potassium channel agonists. Potassium ion channels that we are investigating are voltage-gated ion channels responsible for cell membrane potential homeostasis. It's known that in the therapeutic area that we were interested in, that the potassium channel dysfunction was implicated in the disease state. And that also known was that modulation of this channel was useful for therapeutic indication as well. At the start of the project, we set up some assay screening criteria. We knew that we wanted an assay that was high throughput, which meant that it had to be rapid and easily prosecuted. We also wanted the data to correlate well with manual patch plan. We explored various assay formats that were available, and we ruled out alternative assays that had poor data correlation or assay complexity. In the end, we basically decided that we wanted to run a thallium flux assay. There are multiple thallium flux assays available in the market from different vendors. They have either a wash kit or no wash kits available. At that time, we decided to proceed with the wash version for development. And right at the onset, Catherine from Molecular Devices actually contacted me to beta test their soon-to-be-released no-wash thallium flux assay kit that was in development. I agreed to do so only if it agreed with my timelines. Very quickly, looking at the wash assay principle, it's you load the dye into the cell cytoplasm, you wash off the excess extracellular thallium dye, and then you add the extracellular thallium. Shown here, when the ion channel is closed and you add the thallium and then thallium can't get in, you basically have no reaction with the dye. Whereas on the right side here, when the thallium can permeate the cell, it reacts with the dye, giving fluorescence. This is our final assay wash protocol. This is a fairly straightforward protocol. After plating your cells and incubating them overnight, roughly around 17 to 20 hours, when you're ready to start your assay, you remove the media from your plated cells. You dye load and you incubate for 90 minutes. Inside this dye load, there's added probenecid, and this is needed because you want to block the efflux transport and make sure that you retain your dye. You remove the dye and you wash it with heat buffered HBSS, and when you're ready to start the actual measurements, you add your 20 microliters of assay buffer, you add your compound of interest, you do a pre-read, and this pre-read is to basically identify any fluorescent compounds, and you incubate that for about 20 minutes. And when you're finally ready to read on the Tetra system, you add your thallium. I'm not going to bore everybody with the mundane aspects of this assay, but rather focus on some of what we found the most interesting and key to making sure that our agonist assay worked. And this is the concentration of thallium and potassium is critical in making sure that your agonist assay works. Before looking at some of the critical results, I want to mention how I handled some of the raw data. This is a typical plate setup where you have the 384 well plate designated here with our various concentrations, or our various conditions rather. And right here is the window that shows the typical well signal output. And with these highlighted wells here, it's showing two types of cells. Our stably transfected cell line expressing the potassium channel, and its parental, non-transfected, non-expressing, not overly expressing cell line. Also in here is two different concentrations of opener, and this opener we're going to call opener O. And also, when you see here is that when you add the 2 millimolar thallium, and also in this case, there's actually no added potassium, and you add 80, in the presence of 80 micromolar opener, you see that the potassium channel expressing hex cell shows fluorescence. When you don't add any of the opener, shown here in green, you basically see this baseline level of fluorescence. With the non-transfected parental cells, whether you add the 80 micromolar or 0 micromolar opener, you basically see this baseline. This is to be expected. This is what we saw. So in handling the data, after you define your negative control in green here, you make sure you subtract that out and calculate response over baseline, which is what we did, and we expressed it as either fold or percentage over baseline. And when you do that, this is what you'll see. This is your characteristic signal trace, shown with the 80 micromolar compound and the potassium channel expressing hex cells. Everything else basically looks like a flat line. And then we analyze the max minus min. So this shows the data analysis for the opener O that I spoke about previously. Shown here, there's the parental cell line and the potassium channel expressing cell line. 4 different concentrations of the opener in micromolar. On the y-axis is fold over baseline. And then the 4 different conditions having 4 different concentrations of potassium, 0, 2.5, 5, and 10. And what you see is that the 0 millimolar potassium gives us the best signal to background window to evaluate potassium channel openers, shown here in the dark blue. Also, what you see is that the parental cells actually show very little response to the opener. And whatever response you see is essentially just the basal level of expression of whatever potassium channels are there. On the other hand, the potassium channels that are expressed in this cell line respond to the various concentrations of opener. So you see this concentration-dependent response. Further investigating the potassium effect on the assay readout showed that as you increase potassium, shown here where we basically went from 0 to 2.5 millimolar potassium, your signal to background actually decreases. And when you look at the EC50s, shown on the right here, you notice that as you increase the potassium, you actually decrease the EC50 value, thus increasing the potency. From literature and from our own experiments in the manual patch clamp, we knew that this compound had a EC50 roughly around 10 micromolar. Certainly with these 3 conditions here, we were within the range of expectation. In a similar study, this time varying thallium concentration from 0.5 to 4 millimolar, we saw that the 2 millimolar thallium is the concentration that gave us optimal signals of background, shown here again in green. And when you have very little potassium, such as 0.5 millimolar, you'll see a very depressed signal. So there's actually not much signal to be measured. On the other hand, when you add the 4 millimolar, you see a depression in the signal to background. And when you look at EC50s, you'll notice that as you increase the amount of thallium, the EC50 actually decreases. Honing in, once again, looking at the compound of interest, opener O, the condition where 2 millimolar is present, it is within the range of expectation of manual patch clamp. So, in this bivariate experiment, we were aiming to basically confirm the sweet spot where we have the maximum signal to background and EC50s values that were similar to manual patch clamp. We had 3 different concentrations of thallium, 1, 2, and 3, and 3 different concentrations of potassium, 0, 1.25, and 2.5. And once again, what we saw was signal to background was best when there was 2 millimolar thallium and no potassium present, shown again in green. And then once again, we also saw that when you increase potassium concentrations, you see depression in that signal to background. Moreover, you see the EC50 is most similar to manual patch clamp when you have 2 millimolar thallium once again and no potassium. So at this point, we're ready to look at some reference compounds that were commercially available. In this slide, I show results for 2 of those reference compounds, both of them being potassium channel openers. Once again, opener O and opener P. Setups similar in terms of the experiment, varying concentrations of potassium. And what you see once again is what we saw previously, in that you have the best signal to background at 2 and 0, and as you increase the potassium, you see again the depression of signal to background. More importantly, in both of these cases, as well as another opener, we saw that their EC50s were comparable to the manual patch clamp values. With opener O, the experiment here shows a result of essentially 11, 9, and 11 again. We also tested another compound with the same therapeutic indication, but working by a different mechanism. It didn't work by opening the potassium channel, and we saw essentially no effect. What you saw was essentially for all concentrations, something similar to this red line here. We also looked at 2 potassium channel closers, and it as well showed no EC50 values. So at this point, we had an assay that we felt we were very comfortable with to move forward. But at this point, Molecular Devices actually came to me and had their beta test kit ready to evaluate. I'm going to spend a little time talking about this. The key difference of this kit is a no-wash kit, which means it has an extracellular masking dye to quench fluorescence when your dye actually reacts with the thallium. So, in effect, all you're able to measure what is the fluorescence generated in the cytoplasm. So this is data comparing the 2 different kits. Right off the bat, as I mentioned before, the quencher basically allows you to remove this washing step. And that simplifies assay prosecution. Certainly within our HTS format, it made some of the logistics much simpler and prosecution, again, quicker. The other thing you'll notice is that there's an improved signal to background. With the thallium wash assay kit, you see you get about a 2-fold here, whereas in the Molecular Devices kit, you actually see something very close to 3-fold. I should also point out that I actually did no optimization on this Molecular Devices kit because I didn't have enough beta reagents to do this. All I did was I took the reagent that they gave me and implemented in the optimized assay conditions that I established for the wash assay. In addition to easier prosecution and improved signal to background, we saw also reduced well-to-well variation, as shown here by some of these error bars. But more importantly, we saw that the same compounds are active in both thallium flux assays. When you look at the active compounds B, G, I, M, O, and P, the O and P are the same compounds that we tested previously. We see that the EC50 results are very comparable. And more importantly, both assays were comparable to the manual patch clamp. But given all the other things that we said, the simplicity and the improved signal to background and the reduced well-to-well variation, we decided that the Molecular Devices kit actually had better performance. And we moved forward with that with our HTS. And we had some interesting data from that as well. Unfortunately, the program for that ended, and I don't have much more data on that to show you. So in conclusion, both FLIPR thallium flux assay kits provided functional measurements of the potassium channel openers. Certainly both were within 3-fold of manual patch clamp data. But again, we decided to go forward with the Molecular Devices wash kit because of the simplification of the protocol and assay prosecution. And it saved time and potential points of error. The Molecular Devices no wash kit also improved assay performance by increasing our signal to background, reducing well-to-well variation, and improving data quality overall. I want to take this time to acknowledge the people involved in this project. First, two from Cubist, Hao Chen and Jan, who were the project leads in this project. Keith Russell, who is the electrophysiologist assigned to the project. His manager, Jeff, who was insightful in his knowledge and experience with dealing and working with ion channels. My manager, Deb, and Rory Curtis, my director, for supporting us in our project and making sure that we had everything in place to prosecute. Kristen, who was also involved in, who was involved immensely through her expertise with the FLIPRTETRA, and Tom for his electrophysiology and cell biology experience. I want to acknowledge some people from Molecular Devices as well. Catherine Parrish, who was the first person to contact me with this data test. She was instrumental because she's always involved in keeping up to date with our project needs and our reagent needs. Evan, who contacted us and sort of made sure that we had pre-release of the actual kit, all the reagents that we needed to actually prosecute our HTX. His team worked very diligently to make sure that we got that stuff ahead of schedule. Yen Wen, who worked with me on some of the slides and for the poster. And Xin for setting up this webinar. Thank you all for your attention, and I look forward to your questions.
So welcome to my webinar. As anticipated, I will talk about the setup of the optogenetic assay for the K1 and 3 calcium channels by the use of FLIPRTETRA instrument. And I will touch some different topics in my talk. I will start with a very brief introduction of the optogenetic technique, just to understand which are the potentiality of this technique. In particular, speaking about light actuators and light sensors. And then I will speak about possible applications. Some of them are already being validated, and the possibility to use the FLIPR with this technology. Then I will show you how we developed an optogenetic assay for the K1 and 3 calcium channel by the use of channel adopting tool, and how we were able to set up the protocol to detect state-dependent blockers for these targets. And then I will try to conclude with some of the main achievements of this work and some perspectives for the future. So I would like to start my talk with this comment I found in the web, which is showing how optogenetic technology started entering into the scientific world. More than 20 years ago, scientists discovered that unicellular organisms can change their function upon light stimulation. In particular, they do express ion channel protein, which can be activated by light, and they can induce protons or ion flux in them. So they thought this might be a very interesting technique, in particular for the modulation of, for example, electrical activity of neuronal cells. So you can imagine you can express this protein in a specific cell or cell compartment, and if you are able to stimulate with a light source, you would get a very nice control of the electrical activity. And really, these ones were the first experiments where optogenetics was shining the light in the mysteries of the brain. And now going a little bit more into the scientific part, I just want to remind you that optogenetic technology makes use of genetically encoded elements which are modulated by light. So they are mainly expressed in unicellular organisms. They very often have very simple structure, but most of them can be very well expressed also heterologically in mammalian cell lines. And so, due to the fact that they are genetically encoded, you can express in a specific cell or in a cell compartment. So you can achieve a very high degree of spatial resolution, and you can also modulate them by light stimulation. So you can switch on and off your tool in a millisecond timescale, which is very relevant for biological processes. And all these concepts of spatial resolution and temporal resolution are something that you couldn't achieve by drug stimulation, for example. And as I told you, the first applications were in the study of neural circuits in behaving mouse brain. And but due to the very big advantages and potentiality of this technology, Optogenetics has been nominated Method of the Year in 2010. So now I just want to summarize which are the main optical tools which have been described and validated so far. In particular, we have many optical tools which are also called light actuators. They are proteins which can be activated by light and then they can induce a biological response. Then we also have proteins which can sense and measure a cellular perturbation and they can change their light emission. So, for example, there are proteins which are activated by light and induce function, for example, an ion transport, an enzymatic activity, or a G-protein couple receptor signaling. If you want to study, for example, protein-protein interaction mechanism or translocation, you can use proteins which are activated by light, for example, the Crichton one or the UBR8 or the DRAMPA element. All these proteins encode for light-activated translocation mechanism. You can track your gene of interest activity. You can uncage your activity always by the control of a light stimulation. There are also a growing family of ligands, which can be actually activated in a very fast and reversible way by light. They are called photochromic ligands, and they can also be tethered to a specific receptor of interest. And the last family of optical tools, let's say, are genetically encoded sensors. These proteins are genetically encoded, and they can sense changes in a cellular environment. For example, we have membrane voltage sensors, and we have calcium sensors. Some of them are very well known, like the GCAL. All these tools are very important because due to the fact that they can be, for example, activated by light, you could achieve a degree of precision of stimulation that you couldn't achieve with any drug or chemicals. And because you can deliver the light stimulation in a very fast way, you can do it in a reversible way. And this is something really very powerful. And the optical tool that I will show you today belongs to a channel adopting family, so an ion channel protein activated by light. Most of these optical tools have been already very well validated at different levels. For example, many opsins have been well characterized in vitro, also photoswitchable tethered ligand receptors, and also many genetically encoded indicators. I have to say that most of these experiments are performed with very complex instrumentation with very high power of light source. Then about the in vivo application, as I told you, these were the main application of the optogenetic technologies, where opsin, sensor, enzymes, and very complex mechanisms have been very well validated in animal studies. There is also a very interesting and growing field in ex vivo application, where the optogenetic manipulation of, for example, neuronal cells seems to be very effective in the treatment of some CNS disease. While concerning the drug discovery, optogenetic technology has not been applied very much until now. So we decided to investigate a little bit this part. And as we mainly use the FLIPRTETRA system as a screening instrumentation in-house, we decided to investigate if optogenetic technology would be suitable to be used with the FLIPR system. So probably you will know very well this instrument, but with this instrument we have in your hand an instrumentation which can read in a miniaturized 384-well format. You can simultaneously simulate and measure different wavelengths of fluorescence. Also, you can run multi-mode experiments, so the simultaneous measurement of different wavelengths. You can play with different intensity, duration, or interval of your light stimulation and measurement. And of course, all the 384 wells are measured at the same time. So it would be useful to apply the advantages of optogenetic technology because you could achieve, for example, the simulation only of your optical tool without altering other cells or compartments. With the FLIPR, you have the possibility to run multi-parametric experiments, but it's very important that by using light, you can deliver very fast and reversible stimulus. So, of course, there are a lot of questions about that. For example, you need to demonstrate that your optical tool is well expressed in your cell lines. And of course, we need to demonstrate and to understand if the FLIPR instrumentation, for example, has the suitable wavelengths to study our optical tool, and also if the light power of the LED source was enough. So, the first project that we developed was using channel reduction tool, exactly the Newton D156A. This is a light-activated ion channel, and so upon stimulation with blue light, these opsin transmembrane proteins, which uses all-trans-retinal as cofactor, rapidly translocate cations into the cells, mainly sodium ions and also potassium. It has a very extended open state, so the half decay time of this ion channel is about 7 minutes. So this will induce a very sustained and prolonged manual depolarization. So we thought to use this depolarizing tool to induce a depolarization to activate the voltage-gated Cav1.3 calcium channel. You know, this is an L-type calcium channel, which is a very important CNS therapeutic target. And it's very important for the drug screening of this target to find compounds which are selective over the Cav1.2, for example, cardiac form, but it's also very important to find compounds which are more active on the pathological state of the channel. In the cartoon, in the lower part of the slide, I just show you a reminder of the functioning of the channel. The channel is cycling between different physical states. When the membrane voltage is low, the channel is closed. Then when you have depolarization, the channel opens, allowing the calcium ions to influx into the cells. Then it rapidly undergoes into this inactivated form and then needs to cycle back to the closed state to be able to open again. So state-dependent blockers are molecules which preferentially bind the inactive state of the channel. And there are some possible issues in setting up this kind of assay. For example, we need to know if the channel reduction can be well expressed recombinantly, and of course, we don't know if the LED intensity of the FLIPR is enough to deliver the good blue light stimulation. We don't know if the depolarization will be enough to activate the Cav, and also we need to set up a state-dependent protocol for this target. So this is how we set up our Cav1.3 recombinant assay. So we permanently transfected HEC293 cells with the Cav1.3, the core subunits and also the accessory subunits. And we detected the calcium influx by the use of the calcium-6 molecular devices dye at the FLIPR system. So these cell lines also permanently express the Chiu-Fai Man rectifier potassium channel. This channel is expressed in the cells to lower the resting potential of the cell line, thus avoiding the activation of Cav1.3. So this Chiu-Fai channel can also be used to modulate the Cav1.3 channel by using a classical potassium protocol, let's say, approach. So when the cells are best in low extracellular potassium, the membrane voltage will be very low and the Cav1.3 channel will be in a resting state. If you increase the extracellular potassium concentration, the Q2.3 will allow potassium to influx and to depolarize the cell, so the K1.3 channel will be activated. We express in this cell line also our optically activated depolarizing tool, so the channel rhodopsin that I showed you before. So our goal was to set up an optogenetic assay where when the cells are kept in the dark, the membrane voltage will be at the resting value, so we will have a resting condition. While after blue light stimulation delivered by FLIPR, we would get a depolarization, and so we can modulate the activity of K1.3 channel. The first experiments were, of course, to demonstrate if the blue light of the FLIPR was able to activate the channel rhodopsin and so to induce a depolarization, a sustained depolarization as expected from this channel rhodopsin. So the protocol that we use, we stimulate with the blue light the channel rhodopsin, and we simultaneously measure the membrane depolarization as a fluorescent signal to the membrane potential fluorescent dye. In this panel, you can see that when we apply this first blue light stimulation, the cell responds with a very robust and sustained membrane depolarization. You can see here the increasing fluorescence of the membrane potential sensitive dye. And we also characterize the kinetic profile of this depolarization because we also measure over time the value of the fluorescence of the membrane potential dye. For example, after 5, 10, 15, 20, and 30 minutes after the first blue light stimulation, you can see that the membrane voltage progressively repolarized, and this was consistent with the kinetic properties of the channel rhodopsin. So these experiments demonstrate us that, first of all, we could achieve a good expression of the channel rhodopsin in our assay, and also, very important thing, the LED was strong enough to activate this channel rhodopsin. We could measure a very robust depolarization, and also the repolarization kinetic was very in good agreement with what expected with this channel rhodopsin. So we went on with the characterization of the assay, and so we want to determine if the blue light-induced depolarization was also able to activate the calcium influx to the K1.3. This cartoon is just to remind you the cycling of the K1.3 channel between the different states. So when we applied a blue light stimulation to activate the channel rhodopsin, and this was used simultaneously to measure the fluorescence of the calcium-6 fluorescent dye. And you can see in this panel that the blue light stimulation induced a very strong and robust calcium signal into the cells expressing the K1.3 target. So then we set up a protocol for the study of K1.3 channel in different states. And we achieved this by repetitive blue light stimulation of our cells. As you know, when K1.3 channel opens, it rapidly undergoes into an inactivated state. So what we did is a repetitive light stimulation to follow the time course of the recovery of K1.3 channels from inactivation. You can see in the right low panel, if you immediately repass the same place, for example, after 5 minutes, the depolarization will still be on. So the K1.3 channel will be still in inactivated form. But if you wait a certain amount of time, you can do it in different plates. For example, if you repass your plate 10 minutes after the first stimulation, you can see that about 50% of the K1.3 channel are still in inactivated form. And this was important because by this way, we could set up a condition when we can study the channel in a resting condition. So, I mean, in the first blue light stimulation, or in the half inactivated condition. I mean, if you repass your plate after 10 minutes. So these experiments could demonstrate that the channel rhodopsin depolarization was very well activated in the K1.3 signal. And also, we could also set up a protocol of repetitive light stimulation to study state-dependent blockers. So what we did next is we validated our optical assay with some known state-dependent blockers. I just shown you here the interacting blocker, which is a non-state-dependent blocker. And if we run the light protocol, we can determine the dose response curve and the IC50 of this blocker, either in the resting state, so by stimulating the cells with this blue light, or also we can study the pharmacology in the half-inactivated state by reversing the cells after 10 minutes. And you can see that we could measure very well the IC50 leftward shift of this compound as expected from literature. Then we completely validate our assay with different platforms. For example, with batch clamp analysis, you can clamp the cells at different membrane voltage. You can previously determine in the right panel, you can see the half maximal inactivation potential of this cell line. So you can run exotic pharmacology either in resting condition, for example, clamping the cells at the minus 90 millivolt potential, or in partially inactivated condition, for example, by clamping the cells at the minus 60 millivolt condition, which is very similar to the membrane voltage of pathological neurons. And also in this case, we could obtain a very similar lecture IC50 to what obtained with the optogenetic assay. We also completely characterize the assay by the use of the classical potassium protocol, so by using the tier to modulate the membrane voltage and the channel states. In the low right panel, you can see that we first determine the concentration of potassium in which about 50% of the K1.3 channels are in an inactivated state. And then on the left part of the graph, you can see that this radical dose response curve can be also measured with the potassium protocol. If you load, for example, the cells in physiological 4 millimolar potassium, you will obtain a resting condition of the channel. While if you load in high potassium concentration, you can appreciate the electron shift of the compound also by using this protocol. And so the main conclusion of this work was that channel reduction tool was suitable to set up a very robust and reliable assay for the K1.3 channel and also for the detection of state-dependent blocker of this target. So which are the main differences between running an optical or a classical high throughput screening for such target like K1.3? As a stimulus, when you're running a classical protocol, you are modulating extracellular potassium concentration. This might influence some other cellular processes. And of course, this is not reversible because once you inject your potassium into the cells, you cannot take out, of course. When you are running an optical assay, you are using the light as a stimulus. So you can imagine with the light, you are just activating your optical tool. So these might have really less off-target effects. And of course, the light stimulation can be fast and also reversible. So you can play with repetitive light stimulation as we did in our protocol. And this means also that you don't need to inject any compound. And for example, to run a state dependency protocol with the classical approach, what you usually do is using different potassium concentrations in different places. In these experiments with the optical assay, we use repetitive light stimulation on the same places. And so I have to say also that we validated this assay in a real HTS format, let's say, and it shows very good performance in a pilot screening that we did. So let me give you some possible future perspective of the use of optogenetic technology at the FLIPR or in the drug screening process in general. For sure, one of the most interesting future perspectives would be the use of fast kinetic channel reductions. So I mean, channel reductions which are upon light activation can open and close very, very fast. This would be very useful too because you can imagine you can stimulate electrically yourself in a very precise way with different frequency, duration, and this would be very, very similar to an electrophysiology instrumentation, but in a completely HTS format. Another possible application would be, for example, the use of some of the other options, anion or proton conducting, to obtain really a full control of the membrane voltage, or, for example, also to induce pH modifications into the cells. And also other photoregulated tools like enzyme, or last but not least, the development of assays which are expressing genetically encoded sensor would be very useful to obtain a genetically targeted full optical HTS. And actually, we have ongoing a lot of activities in many different projects in this field. So we hope to have some new results soon to share with you. Just as a conclusion, what are the main messages that we can take away and use from this work? The first one is that the channel dot seems show a very good functionality at the FLIPR, so this is an interesting optical tool that can be used at the FLIPR. The second message is that we were able to develop a stable HTS-compatible assay for the K1.3 target by the use of channel dots in optical stimulation. But I think that probably the most relevant message is that we can think about using the FLIPR for running light-modulated high-throughput screening, and this will really open the biggest possibility for the futures. And just let me finish with the acknowledgement of all my colleagues who contributed to this work, and I'm happy to take any questions.